Hi everyone. I hope I'm audible. So, just excuse my voice. I'm having a little cold and fever a bit, and that's the reasons this is low. Okay. So let's get started. So before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Okay, I'm visible, right? All right. So join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology. So this you can just take it as 9 and 10 Biology. And the Telegram channel of Unacademy, ICSE and IC both. So this link you can find in the description. Okay. So in description you will find the link for sorry, so you'll find the link for an academy channel, and also you find the link for community, the special classes, and also you will be finding a link for the bucks bounty as well. Okay, so you can just go to the description and find out the links there. So we'll be conducting special classes on an academy platform. All you need to do is download the Unacademy Learners app and learn from your favorite educator for free, because the special classes are basically the free classes which are conducted on the platform, that is an academy platform, where you can engage with the educator via poll, because right now a lot of educators are conducting a quiz. Okay, so you can just take part in them, so clear your doubt in the live special class as well. Now for the top special classes you can visit the link in the description. So we have a option here where we call that a question tab, where you can type in your question, attach an image and send it to the educator during a live session. So the educator will be getting a notification that the learner has asked a doubt and will be clearing your doubt. So this is about an academy subscription features. So you can learn live from the comfort of your home. Unlimited access to all the courses will be there. Then top educators of India are going to take the classes in one platform, that is an academy platform. So here different educators will come up with different courses. So you will have access to all the courses once you take the subscription. And also there is a regular doubt clearing session after about every 3 session. So the fourth session will be your doubt clearing session. So whatever doubts you have related to the topics that are previously covered, so you can clear your doubt in this doubt clearing session. Okay, and complete syllabus will be covered, mentorship and guidance will be given, the study material is given, so that will be uploading in the form of a PDF itself. So you can just download the PDF from the respective class, so you can have your own notes with this. And also we'll be conducting a practice test, live test series are conducted. So we have a batch in English, okay, and we have a batch in English as well. So once you take the subscription, so you will have access to English as well as a English batch. And with that there is daily practice section, weekly mock test series are there, live quizzes are there, daily MCQ and subjective test series are also conducted. So once you go for the subscription, so basically you can take a benefit of all these features. And right now there is a offer also that is going on, which is basically ICSE combo offer for ICSE class 9 as well as for class 10. In the meanwhile you can prepare for JEE as well as NEET. So basically this is 1 year subscription. Along with this you will be getting this subscription as well, where the cost is basically 34,500. So this is for class 9 and class 10, so it is anyway the same thing. So here, the cost has been reduced because of the offer, where it is kept as 31,050, but with the code. So this is my code Vani YT. So with this code you will be getting a discount on this particular offer, so that is basically 10% discount. Okay, and the link for these particular offers, so the link you can again find in the description itself. All right, or you can join in my telegram, that is Vani Kari ICSE class 9 and 10 biology. So you can join in the telegram, so there I have sent the link, so you can take the link from there as well. And this is my referral code Vani YT to get 10% discount on this particular offer, where you can study for class 10, and along with that you can also study for JEE NEET as well. Okay, so right now we have this offer. So this offer we have for a month. Okay, so this offer is gonna exist for a month, but this is gonna end by 30th of June. Okay, so get 20% off on ICSE board plus subscription, where for 12 months it was earlier 17,250, but till the 30th of June, so you have this offer where you will be paying just 13,800, and this is the amount that you are saving. So this is with the 20% offer. Okay, so you can use the code Vani YT again to get this offer. So this is the code Vani YT to get 20% off. For 12 months it is not 17,250, it is 13,800, and this offer is going on only till 30th. This is valid, and after that so again the normal thing, basically 17,250. So you can use my code Vani YT to get 20% discount on this offer. So this is the regular one which will be there. So for 12 months it is 17,250. Since right now there is offer going on, so you can avail the benefit of this one. So we'll study about the structure of DNA today. I'm really sorry about my voice. So I'm having a cough and a little fever, and that's the reason. Okay, so moving on to introduction, so that is basically related to the DNA. So since we are going to discuss about the structure of DNA here, okay, so this is about the structure of DNA. So how exactly formation of DNA occurs, what is the DNA content, and where exactly this DNA is present, what is the role of this DNA. So we'll see that, and before that we'll discuss about how the arrangement is done in the DNA. So DNA is the basic genetic material which is required and which has been transferring from one generation to the another generation. So now the younger generation or the progeny which is produced, so that is having a DNA both from mother as well as father. Okay, so both of them are passing their DNA to the child, and now this child, when that child grows up and that gets married, so the women that the child, I mean the adult is getting married, and again it gets, I mean it gives rise to a progeny, right? So again it will have a combination of these grandparents, because already the parental DNA is from the mother side, the grandparental, so that DNA. So basically generation after generation, it's not that only the parental genes are being expressed, it is basically the genes we are obtaining or the DNA that we are obtaining, so it is coming from generation to the generation. So flow of DNA is occurring, and that itself we call as a heredity or inheritance. When we mention, basically it is DNA passing from generation to the generation. So here we just mention about the parents. So if we go even for a higher study, so it can be the grandparents, the great grandparents, so from there the DNA is coming, right? So DNA is the genetic material that is for most living organism. So how does the length of the DNA fit within the space of a nucleus. So let's say this is a cell now, okay, and this is this particular nucleus. So what is this cell actually? So this is not even visible to our eye, okay. So that means you need a microscope to observe a cell. So it is that tiny cell which is not visible through the naked eye. So you cannot see it through the naked eye. And you know how much of DNA is there in each cell? So that is about 2.2 meters of DNA you can find. So what is 2.2 meter? Like this huge, okay. So this is just 1 meter. So as such, basically, like a big one if we consider. So basically 2.2 meters. So such a big DNA is present. So I think you can imagine 2.2 meter. So in that case, so such a big DNA is fitting into that tiny cell, which is not even visible through our naked eye, and not just fitting in the cell, but it is fitting inside that tiny nucleus which is present in that tiny cell. So that means such a big DNA has to get fit into this nucleus, right? Basically, let's say this is DNA. So this is the DNA, separated DNA strand. So now in that case, you can see that the DNA is going to form a copy of itself here. So this is going to form a copy of itself here. So that is a replication, basically duplication, or we can also say that the semi-conservative replication has occurred. So that means DNA is getting multiplied. So let's see this one today. That is how does such a big DNA is fitting into this tiny cell nucleus, okay. So you know what exactly will be there in a nucleus when we mention. So we'll talk about the nucleus first before we study in detail about what exactly is DNA. One second. Yeah. So now alright. So now nucleus is basically covered by a nuclear envelope. So what does this nuclear envelope contain? So nuclear envelope, when we mention, so nuclear envelope is having the outer membrane as such. So there is the outer membrane. So this is only, I'm drawing only the nucleus, not the whole cell. So this is the outer membrane here, okay. And then there is the inner membrane. So this one is the inner membrane. So this one will form a nuclear envelope along with the nuclear pore. So what is this gap now? So this gap is basically a nuclear pore. So these are the gaps, okay. So this outer membrane and the inner membrane will join together, and this we call as the nuclear envelope. And there you can see a nuclear membrane. So this is now a nuclear membrane. So we call this DNA is covered by a nuclear membrane, right? So means what? So there is a well defined nucleus. Why do we call this as a well defined nucleus? Any idea? Why do we say that this is a well defined nucleus? Because there is a membrane. So there is a outer membrane, there is a inner membrane, and within this structure itself, so now this is basically a nuclear pore, okay. So this is a nuclear pore, and you can see the outer membrane, you can see the inner membrane there, all right. So now with respect to this, here inside that you can find a nucleolus, okay. So there is a nucleolus, and here you can find the chromatin fiber. So chromatin fiber is basically randomly arranged structure, a thread like structure that you can find in the nucleoplasm. So here a nucleoplasm will be there. So I'll just highlight that, okay, just a second, so that you can understand where exactly, what exactly is nucleoplasm here. So this part you see here, right? So this is completely a nucleoplasm itself, okay. So all this space what you see here, so nucleoplasm is in continuity with the cytoplasm actually, because surrounding you can find a cytoplasm, okay, and it is in continuity with the nucleoplasm here. So that is because through the nuclear pore. And now this structure, what you see, so that is basically a chromatin fiber. So this one is what? This is a chromatin fiber, and this chromatin fiber, when we mention, so this is basically the one which is condensing to form a chromosome. So I'll explain you about this. Okay, so is this clear about nuclear envelope? Nuclear envelope, when we mention, the nucleus is bound by two membranes that together forms the nuclear envelope. So this will be porous because there is a outer membrane, because there is a inner membrane, so both of them join together, and there is a pore formation. So nuclear pore is also there. Okay, so nucleoplasm is basically the liquid that you find inside this nucleus. So this is basically nucleus here, this is completely nucleoplasm will be there. This is a nucleolus, and this one is basically the nuclear pore, and some attached ribosomes also you can see on top here. How many strands make up a DNA double helix? So it is only two. Okay, so two strands of a DNA will be there. So I'll just draw it here. Here, okay. So this is one strand of a DNA. There is another strand of a DNA. So two strands of a DNA are gonna make up the DNA helix there. Okay. Yeah. So now with respect to that, the nucleoplasm is present, so which is a liquid region inside the nucleus. And so this contains nucleolus and chromatin. Now we'll study about this chromatin. What exactly is chromatin? So this is the thread-like mass which you find inside the nucleoplasm, that is inside the nucleus. Okay, and this consists of DNA. So there is a DNA and then there is a protein that you can see. So DNA and protein is present. So that means a nucleosome model. So I'll just explain you what exactly is nucleosome model. So this chromatin consists of a nucleosome model which consists of the DNA, and also there are proteins associated with this. So this carries the genetic information because we have obtained this DNA from the parents. So nucleolus, when we mention, maybe one or many can be them. So synthesis of ribosome is the function of this one. So this is the histone bead. Okay, so that is bead on a string we call, because surrounding this bead you can see there is a DNA which will be there. Okay, so let's see, this is now a DNA which is coiled around this. So this is a DNA, and this histone bead, what you see, so this is actually the protein here. Okay, just a second, I'll drink some water. Yeah, so now this we call, this itself we call as a nucleosome, because nucleosome, no, it is having a DNA, all right, and also it is having a protein. So now when we mention about the chromatin, so as such you can find many units of this nucleosome. Okay, so many units of nucleosome where you can see the DNA is coiled around, then you can see this is being formed, so this is being formed, and this is being formed there. Okay, so this is a nucleosome model you can see here. So this is one nucleosome, this is one nucleosome, and this is one nucleosome. So all of these are basically nucleosomes itself. So many units of such nucleosomes which are there. So these are going to form that chromatin fiber. Okay, so this is the one we call as a chromatin fiber, which consists of the DNA and then the protein. So basically many units of nucleosome. So now you can see there is a chromatin fiber, which is nothing but the many units of nucleosomes can be found in there. Okay, and now while the cell is going for the cell division, so this cell is now undergoing a cell division. So as the cell is getting ready for cell division, so you can see that the chromatin fiber will start to condense. Okay, condenses basically all of them are coming closer. So there is a coiling and supercoiling that can be seen. Okay, so coiling of a DNA and supercoiling of a DNA you can find. So that is to arrange itself as the chromosome then. So chromatin fiber will arrange itself. So I'll just write here. Yeah, so now the chromatin fiber what we mentioned. So chromatin fiber is going to arrange itself as chromosome, that is by coiling and supercoiling, so which we call as the condensation. Okay, any doubt in this? So this is basically a condensation process. Okay, so that means the chromatin fiber, when we mention this itself, is having many units of nucleosome. So these many units of nucleosome what you find, so all those are basically they are coiling, they are supercoiling. So what is coiling and supercoiling? So now and this we call as coiling. Okay, so this is coiling, and then you can see there is a supercoiling as such. Okay, so this one is coiling and then there is supercoiling. So coiling is this, supercoiling is this, coiling is this, and there is supercoiling is this one. All right, so coiling and supercoiling can be seen, where you can see that the chromatin fiber is arranging itself as a chromosome, that is by condensation, and where the condensation is going to occur by coiling and supercoiling. So this is the one here where you can see there is a DNA. Okay, so this DNA is coiled around that protein. So this is a protein now. So as such you can find many such proteins. So there are many such histone proteins are there where you can see the coiling. Okay, so this is now a histone protein. So this structure itself is gonna coil. Okay, so this whole structure you can see the coiling over here, the coiling has made it to be the coiling here, and then you can see this is gonna be the super coiling now. Again there is coiling, super coiling. So this way the whole of a nucleosome in a chromatin fiber. Okay, so this is in a nucleosome in a chromatin fiber. Excuse me. Okay, so I'm having cold, little excuse me. So this is basically nucleosome is gonna arrange itself as the chromosome then. So here in that case this is a chromatin fiber which will arrange itself as a nucleosome. Later basically there is a coiling and then you can see formation of this chromosome. So if you just try to pull out the thread from the chromosome. So it will be of a chromatin fiber which is coiled, which is supercoiled, and you can find further separation. You can find the nucleosomes clearly. Further separation, you can find 8 histone cores, that is to form the DNA. So let's have a look at the DNA packaging in case of eukaryotes, and then I'll explain about what all will be there in this DNA. Okay. So now with respect to DNA packaging in eukaryotes, so basically you can see that one nucleosome will have about 200 base pair. So what exactly is the base pairing now? Anybody has any idea what is this base pair? When do we say base pairing of a DNA? So there are about 200 base pairs of a DNA helix. So nucleosome repeat in a thread-like structure, so that we call as a chromatin, which is present in the nucleus. So now further supercoiling of chromatin creates a loop structure of chromatin fiber and then the chromatid. So this structure condenses at the metaphase of the cell division to form a chromosome. So basically condensation will start in the prophase itself. So condensation is going to complete at the metaphase. So here in this case, let's see the structure of DNA. Okay, so I'll just explain you the structure of DNA. One second. So DNA is what? Anybody? DNA is basically, it is a polynucleotide chain. Okay. So what is it? It is a polynucleotide chain it is. So what does this polynucleotide chain consist of? That means many nucleo. Poly is meaning of poly is what? Meaning of poly is many. Okay. So many nucleotides are there. So what exactly is a nucleotide then? So nucleotide, when we mention, so nucleotide is made up of the nitrogenous bases. Okay, so there are nitrogenous bases that you can find. Then also you can find there is pentose sugar. Okay, so there is a pentose sugar that can be found. Then you can also find that there is a phosphate group. So all these are the components that you can find in a nucleotide. Okay, so nucleoside is also there. I'll just write it here. So nucleoside is nothing but without the DNA, I mean without the phosphate. So this nitrogenous base plus pentose sugar is gonna form the nucleoside. So nucleotide, it is a different one extra group, so that is from nucleoside. So which we call as a nitrogenous base, pentose sugar, and then there is a phosphate group that can be formed. Or so some like a few people can write the nucleotide to be. So nucleotide is equal to nucleoside plus phosphate also they can write, because phosphate group is not there in the nucleoside, right? and hence we can write this equation as well, or this is the equation that you can go with, or this is the equation that you can go with. Yes, any doubt? Is it visible? So now the structure of DNA, will discuss about that. So what is the total length of a DNA that a human body may have? Any idea on this? Any of you can just type in. So it is approximately, you can find about 110 billion miles. So that is the amount of total DNA that you can find. So Ankesh has answered don't know. Okay. So if one DNA strand, so this is per cell. Okay. So in each cell, I told you guys that there is about 2.2 meter of a DNA which is compactly arranged here in the nucleus basically. Okay. So that means now we have about trillions of cells. Okay. And for that trillions of cells, so now this is the question is, what is the total length of a DNA that a human body may have? So since we have trillions of cells together, so basically it is about 110 billion miles. So if one strand is about 2.2 meter, the DNA from the 100 trillion cells in the body put end to end, so it would take us to the sun and back about 600 times. So you can see that the DNA is basically in comics, so they have used this as a ladder, because DNA, if you observe, so that anyway looks like a ladder itself. So somewhat like this it will be there, right? So this is the DNA strand that you can find, and you can see there are base pairing. Okay, so there is a base pairing that can be seen, and that's the reason. So this one, and like we cannot literally climb the DNA and go to the sun and come back. So this is just the imagination which can tell you about. So if we just put them end to end, all the DNA from the cell, so in that case we can go to the sun, and also we can come back from there. That is not just one or two times, so that is about 600 times. So that means we have so much of DNA in our body which is carrying information. So where is DNA present in the eukaryotic cell? Anybody? Inside the nucleus with other cellular elements or cellular content, then inside the ribosome, and not at all present. So where exactly do you find this nucleus? I mean, where do you find this DNA? No idea? Okay, so where is DNA present in the eukaryotic cell? So this you can find inside the nucleus, because I already told you there is a nuclear envelope, and this nuclear envelope is basically outer membrane, inner membrane, which are fused together, and there is a nuclear pore here as such. So these are anyway fused, and now so this is the one, and you can basically see this is one second. Yeah. So this is the nuclear pore, where there is a formation of a nuclear pore. So now the DNA is present inside this nucleus, because this one is anyway the nucleus. So the structure what I have drawn is of a nucleus. This is nucleolus. Okay. And inside this nucleus you can find there is a nucleoplasm. So this is completely a nucleoplasm which will be here. So now this nucleoplasm will have a chromatin fiber or the DNA. Okay, so basically DNA suspended here itself, So that is inside this nucleoplasm. So that is in the nucleus. So let's just draw the DNA there. So here you can find a DNA, so which is basically a random thread like structure that you can find inside the cell. So this is what? This is the DNA. So DNA is present in the eukaryotic cell. So that is inside the nucleus. So moving on to next one. Which of the following comes under level of organization of DNA? So nucleosome, chromatin fiber, solenoid, and all of the above. So what is the answer for this? So Shri Krishna has answered this to be A, the previous one. Yes. So what is the answer for this? Just a second. Okay. So which of the following comes under the level of organization of DNA? So this one is basically nucleosome. Then there is chromatin fiber also, because many units of nucleosome itself are forming the chromatin, and this chromatin fiber is coiling and it is supercoiling. So that means it will arrange, like solenoid is what in physics you study, right? So this is the solenoid. So basically the coiled wire, like wire is continuously coiled over there. So there you can see that the chromatin fiber is coiling and it is super coiling as such, which will be like a solenoid. So it is the which of the following comes under the level of organization of DNA? So it is all of the above, because you can see nucleosome, there is a chromatin fiber, and also there is a solenoid. Okay, so in the beads on a string model, the bead is made up of, any idea on this? I'll explain you this. For now, do you guys have any idea what exactly bead is made up of, or bead on a string model? So this can even be called as a nucleosome. Okay, so this can even be called as a nucleosome. No idea? So this is basically 8 histone proteins and a DNA you can find. So I'll just explain you how. Now this is a histone protein, this is a histone protein, there you can find a histone protein, and then there is a histone protein. So I'll just say erase this. Okay, so now histone proteins, when we mention, so you can find many units of histone protein there. So at the back also you can find there is a histone protein which will be present. Okay, so you can find 8 of the histone proteins. So that is H2A, H2A, H2B, H2B on the back. So this is H3 and this is H4. So H3 on the back, H4 on the back. So we can just write it here, H2A, H2B, and here it is H3 and here it is H4. So 8 histone proteins you can find, 1, 2, 3, 4, 5, 6, 7, 8. Okay, and now this is considered, so this is one whole bead itself. Okay, so now coiling this bead, you can find there is a DNA which is coiled around this, and now this is the DNA. Okay, so nucleosome, when we mention, so it is not just the histone protein or not just the DNA, so both of them together. So DNA plus 8 of the histone proteins together, so we call this to be a nucleosome. What is, so 8 histone proteins it is. Alright, so this we call as what? This is a nucleosome model or beads on a string model. So now with respect to that, Just a second. So single human chromosome, we have about million nucleosomes. So there is coiling, there is supercoiling. So concept of coiling and supercoiling. One second. So here it was basically a single human chromosome. So this we are talking about the human chromosome, which is just one single chromosome. Okay. So may have about a million nucleosome. So I just told you that many of the nucleosomes together are going to form a chromatin fiber. So now you know a single human chromosome when we mention. So this will have about million nucleosomes there. Okay. So these million nucleosomes when we mention, so you can see coiling there, you can see supercoiling there. Okay. So coiling and supercoiling is done. So this itself is forming the chromosome. So this anyway they have taken an example of telephone wire. So concept of coiling and supercoiling as is also found in chromatin fiber of the chromosome. So same type you can find, but anyway now here with respect to this, so you can see there is a coiling. So same way in chromatin fiber along with its nucleosome model. So here you know many of the nucleosomes will be there. So with their nucleosome itself they are coiling and they are supercoiling. So DNA packaging in eukaryotes when we mention, so this is about the packaging that you can see. Let me just explain you this. So this is H2A, a histone protein. So histone protein when we classify, so histone protein is classified as H2A. It is H2B and there is H3 and you can find H4 and there is H1. A linker protein will also be there. So here you know the one which is forming the core here. So this is H2B and here you can find H3 and here you can find a H4. So at the back you can find each one of them at the back. So this is gonna be a core protein H2A, H2B, H3 and there is H4. So this is the core. Okay, so this is core of what? Histone core protein, and this histone core protein is basically coiled by a DNA around it. So in that case, so you can see that the DNA is coiled around. So this is a DNA nerve. Okay, and this one is a histone protein. So let's see this. This is a histone core protein. All right. So with respect to that, we can see, you can see there is a packaging which will be done. So such a big DNA is arranged in that tiny place that is inside the nucleus, that is by coiling and supercoiling around the protein. So the proteins in eukaryotes, they acquire negative charge, that is from the amino acid, right? So this is basically, you know, the positive charge we can mention. So there is a positive charge from the amino acid residues with the charge side chain. The amino acids lysine and arginine form histone, a kind of protein. So 8 molecules will be there which are positively charged histone. So they are basically here. The protein is the one that is because of the amino acid lysine and then arginine. So because of these two, so you can see that basically this has got the positive charge. Okay, so in that case the negatively charged DNA, so DNA is the one which is negatively charged and this DNA wraps around the histone octamer forming the nucleosome there. So name the scientist who proposed the nucleosome model. Anybody has any idea? No idea on this? Okay. So Roger de Comberg, he is anyway the Nobel Prize winner. American scientists show that the nucleosome serves as a general gene repressor and prevents the initiation of transcription. It is Don and Ada Overlands were the first to discover the structure of nucleosome. So he anyway got the Nobel Prize, but the discovery about the nucleosome was done by the Don and Ada Overlands. Okay, so here I'll just explain you the structure of DNA. Now in structure of DNA, basically you can find this to have okay, 1 second. So since we said that this is a polynucleotide, so first we'll discuss about the nitrogenous bases. Okay. So there are nitrogenous bases. So nitrogenous bases you can find two types. So there is a purine, there is a pyrimidine. Okay, so there is a purine base you can find and there is a pyrimidine you can find. So now as the name itself suggests, so purine are basically double ring structures. So the name is small but the structure is larger, that is double ring. So in that case it is adenine. Okay, and then there is guanine, and the other one the pyrimidine. So pyrimidine is basically a cytosine. Then you can find a thymine. So these are the structures or the nitrogenous bases which are found here, which we call as ATGC in general, where the A is gonna make a pair with the T, that is by 2 hydrogen bonds, and G is going to make a pair with C, so that is by 3 hydrogen bonds. Okay, so this itself we call as a base pairing. So what is base pairing? So let's say here a DNA chain is there, so I'll just explain it here. So this is the chain of the DNA. Okay, and you can see this is the DNA chain, so we'll just draw them a little further. Okay, so this is now a chain of DNA, and this is the structure. So now let's say here A, T, G, C is here, and the complementary base pairing is gonna be on this side you can find T, on this side you can find A, on this side you can find C, and on this side you can find G. So you can just make a note there. So this is basically, you can see there, 3 of the hydrogen bonds are there between G and C, and 2 hydrogen bonds are there between A and T. So now with respect to that, here also it will be basically like this, the base pairing will be there. So depending on the type of the base pair that is there. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it is basically 10 has to be there. So 10 base pairs will be there for each turn. So based on that, you can see there is a base pairing of a DNA which is done, and this base pairing is basically, this is a double stranded DNA. Okay, so there is a base pairing that can be seen. So now with respect to this base pairing, so now so here we said that this is base pair. Okay, and this strand, what is there? The strand is made of a phosphate group and there is a ribose sugar. So there will be a deoxyribose sugar in case of DNA. So this is basically a deoxyribose sugar in case of DNA, and a phosphate group can be found. So this is in a double stranded DNA. So I'm not visible, right? Okay, just half visible. Yeah. So now the DNA strands, we say that they are complementary. So why do we say that the DNA is complementary to each other? So we say that they are complementary to each other. So that is because the complementary base pairing will be there. So A is always complementary to T, and G is always complementary to C. So G is always going to pair with C. So this we call as a complementary base pairing. Okay, so this is a complementary base pairing where you can see A is pairing with T and G is pairing with C, and hence we say that the DNA is complementary to each other, because Always this is gonna bind with this itself, that is A is gonna bind with T itself, T is gonna bind with A itself, G is gonna bind with C itself, C is gonna bind with G itself. So other than that there wouldn't be any bonding, and two hydrogen bonds has to be there, and that's the reason we say that the DNA is complementary. And now with respect to the other ones, so that is hi Pratham. So with respect to the other one, so quiz pooch lo, I just finished some of the questions. Anyway, so I just finished it, like some of the questions from the previous one. And other than this, so DNA is complementary, and we say that the DNA is anti-parallel. So why do we say that the DNA is anti-parallel in nature? So when we mention that the DNA is anti-parallel, so now let's say one of the strand is running in 3 dash to 5 dash direction, the other strand is basically running in 5 dash to 3 dash direction. So these are the changes. Basically, one of the strand goes in 3 dash to 5 dash here, okay, and the other strand is basically 5 dash to 3 dash. And you can see that even though they are parallel, we say that it is 3 dash to 5 dash and 5 dash to 3 dash. That is because it is anti, so they are running in opposite direction. So if one strands, one second, yeah, so if one strand so just kidding, okay, so one strand runs in this direction, the other strand runs in the other direction. So basically it is 5 dash to 3 dash, the other one here is 5 dash to 3 dash, or you can say 5 prime to 3 prime, and here also it is 5 prime to 3 prime. So they are parallel, but since they are running in opposite direction, so it is a anti-parallel. So that's the reason the DNA is said to be anti-parallel and the DNA is said to be complementary to each other. So some questions I already completed, so you can just answer for this one, Pratham. So in the beads on a string model, that is nucleosome model, the bead is made up of 6 histone protein, 8 histone proteins, 6 histone proteins and the DNA, and 8 histone proteins and the DNA. So what is the answer for this? Any idea? So this itself I explained in today's class, like what all will be there in a nucleosome model. So that is along with that the DNA structure. So this is basically 8 histone proteins and then the DNA can be found in there. So this and all is done. So before I end this session, so let me just say about the batch that is gonna come next in July. So this is a Titans batch, which is basically all subject batch, which will be starting from July 13th, and the mode of delivery here is English. So you can go for maths, physics, chemistry and bio. So if you are subscribing for class 10, so you can basically get the subscription of class 11 and class 12 subscription absolutely free along with this. And there is a combo offer which is going on, 8 histone protein, and then there is a DNA also that is present, because nucleosome model, right? So there is a DNA which is called around this 8 histone protein, and that's the reason 8 histone protein plus DNA it is. Okay, so in that case ICSE combo offer for since you are in class 10 now, so there is a offer going on right now, so where you can start your early preparation for these competitive exams like JEE as well as NEET. So you can take a subscription for class 10, and with that you can also take a subscription of JEE and NEET for this amount, where you can see earlier it is 34,500 and now this the offer is 31,050, and with the referral code Vani YT. So this is my code Vani YT. So you will be getting 10% discount on the subscription. So link for this you can find in the description. Okay, so you are in fifth class. Okay, I didn't know that. Nice. Anyway, you still answered. So now get 20% off on ICSE board. So that is plus subscription. So in that case, so you can get again the same code can be used, that is Vani YT, to get 20% discount on the subscription, where this offer is going to end on 30th of June. So subscribe now using the code Vani YT. So where so let's say you are taking a subscription for 12 months. So instead of 17,250, so you will be just paying 13,800. Okay, so that is basically you need to add the code there, that is 20% discount you will be getting. So introducing community, but all questions because I read the book a lot. Nice, that's good. So introducing community. So never miss on any update from your favorite educator. So get session notes and practice material. Share your feedback directly with the educator via poll. So Bucks Bounty. So this is opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. So I hope this is clear. So all you need to do is download the Unacademy Learners app and start taking up free classes which are available. So if you are comfortable, you can go ahead with the plus classes there. Okay, so all you need to do is download the Unacademy Learners app, sign in and start taking up the session. So subscribe to our channel, that is Unacademy ICSE and ISC boards. Click on bell icon to get notified about the session. Like and share the video. Download the Unacademy Learner app. This PDF, if you need, I can anyway send it on my community or Telegram. So you can join in my Telegram. So see you all in the next session. Bye bye everyone. Take care. Thank you all.